DNA fingerprinting, or DNA profiling, is a groundbreaking technique for identifying individuals by their unique genetic makeup. Unlike traditional fingerprints, DNA fingerprints are derived from our DNA, the building blocks of our body. This tool has revolutionized forensics, law, and medicine, offering unprecedented certainty in identification. It links suspects to crime scenes, establishes family ties, and identifies disaster victims with precision. DNA fingerprints are internal, biological markers, stable throughout life, and inherited from parents. The chance of two unrelated individuals sharing the same DNA profile is astronomically low, making DNA evidence compelling in court. For UPSC aspirants, understanding DNA fingerprinting is crucial for connecting science with governance, law, and ethics. We'll explore its scientific basis, applications, and societal implications essential for the civil services examination. DNA fingerprinting is a remarkable scientific process that hinges on the unique structure of DNA, the molecule that serves as the blueprint for all living things. At its core, DNA is a double helix, a twisted ladder-like structure that contains the genetic instructions necessary for life. This double helix is made up of 4 chemical bases: adenine, thymine, cytosine, and guanine, arranged in a specific sequence that encodes our genetic information. While an astonishing 99.9% of DNA is identical among all humans, it's the remaining 0.1% that accounts for our individual differences, from eye color to susceptibility to certain diseases. This tiny fraction is what makes each person genetically unique, much like a fingerprint. Within this 0.1%, scientists focus on regions containing repeating sequences of DNA, known as variable number tandem repeats, VNTRs, and short tandem repeats, STRs. These repeating patterns are the key to DNA fingerprinting. The number of repeats at each location or locus varies greatly among individuals, creating a pattern as distinctive as a signature or a stutter in a sentence. By analyzing these repeating sequences at multiple loci, scientists can build a unique DNA profile for every person, which is invaluable in forensic investigations. Importantly, STRs are selected from non-coding regions of DNA, areas that do not code for proteins or reveal personal traits, ensuring that privacy is maintained and sensitive genetic information is protected. By examining a combination of several STR loci, forensic scientists can generate a DNA profile that is statistically unique to an individual, making the chances of two unrelated people sharing the same profile astronomically low. This process translates the lengths of STRs into a numerical code, which can then be compared across databases to reliably differentiate between individuals. The use of non-coding DNA for identification is the cornerstone of forensic genetics, allowing experts to link individuals to crime scene evidence with remarkable accuracy. This robust and scientifically sound method forms the very foundation of modern forensic genetics, revolutionizing the way we solve crimes and identify individuals. Transforming a DNA sample from a crime scene into compelling courtroom evidence is a journey that demands precision, expertise, and unwavering attention to detail. Every step, from the initial collection to the final presentation in court, is critical to ensure the integrity of the evidence and the pursuit of justice. The process begins at the crime scene, where forensic experts carefully collect and preserve biological samples such as blood, hair, or skin cells. These materials are highly sensitive, and even the slightest contamination can compromise the entire investigation. That's why strict protocols are followed, using sterile tools and sealed containers to maintain the purity of the evidence. Once the samples arrive at the laboratory, scientists meticulously extract and isolate the DNA from the collected cells. This step involves breaking open the cells and separating the genetic material from other cellular components, ensuring that only the DNA is retained for further analysis. The extracted DNA is often present in tiny amounts, so scientists use a technique called polymerase chain reaction, or PCR. This process acts like a molecular photocopier, amplifying specific segments of DNA millions of times so that there is enough material to analyze accurately. Next, the amplified DNA fragments are separated by size using gel electrophoresis. This technique sorts the DNA based on length, creating a unique pattern of bands that can be visualized and compared. The resulting pattern, often called a DNA fingerprint, is made visible with fluorescent dyes under ultraviolet light. Each person's DNA fingerprint is unique, making it a powerful tool for identification. Forensic analysts then compare the DNA profile from the crime scene with profiles from potential suspects. A match can confirm involvement, while a mismatch can exclude individuals from suspicion, providing clarity in complex investigations. In the courtroom, DNA evidence is presented to judges and juries as a scientifically robust form of proof. Its reliability and objectivity help ensure that verdicts are based on facts, not speculation. Over the years, DNA fingerprinting has played a pivotal role in solving countless cases, exonerating the innocent, and bringing perpetrators to justice. It has revolutionized forensic science and transformed the way crimes are investigated and prosecuted. The journey from crime scene to courtroom is a testament to the power and precision of DNA technology, connecting the smallest traces of evidence to the truth. In the end, the silent language of DNA speaks volumes, ensuring that justice is served and that every story told in the courtroom is grounded in scientific truth. The fascinating story of DNA fingerprinting began in 1984, when British geneticist Sir Alec Jeffreys made a groundbreaking discovery in his laboratory at the University of Leicester. While studying the genetic markers in human DNA, Jeffreys noticed that certain regions of DNA varied greatly between individuals, creating unique patterns that could be used to identify people with remarkable accuracy. This accidental discovery would soon change the world. Jeffreys' breakthrough revolutionized forensic science and justice systems across the globe. For the first time, scientists and law enforcement had a reliable method to match biological evidence to individuals, transforming the way crimes were investigated and solved. DNA fingerprinting quickly became a powerful tool, not just for solving crimes, but also for reuniting families and resolving complex legal disputes. The first legal use of DNA fingerprinting came just 2 years later, in 1986, when it was used to resolve an immigration dispute and a high-profile criminal case in the UK. The technology's ability to provide definitive answers in courtrooms marked a new era for justice, setting a precedent for its use worldwide. In a landmark case, a DNA manhunt led to the capture of Colin Pitchfork, the first criminal convicted using DNA evidence. This case not only demonstrated DNA's power to convict the guilty, but also to exonerate the innocent, forever changing the landscape of criminal investigations. Meanwhile, in India, Dr. Lalji Singh emerged as a pioneer in the field. He introduced DNA fingerprinting to the country, applying it to solve major cases, including the investigation into the assassination of former Prime Minister Rajiv Gandhi. His expertise brought scientific rigor to Indian forensic investigations. Dr. Singh's efforts led to the establishment of DNA evidence as a cornerstone in India's legal system. He founded the Center for DNA Fingerprinting and Diagnostics, which became a hub for research, training, and casework, ensuring that India kept pace with global advancements in forensic science. Thanks to early adoption and visionary leadership, India quickly became a leader in forensic science, using DNA technology to solve complex cases and deliver justice more effectively. Pioneers like Sir Alec Jeffreys and Dr. Lalji Singh transformed DNA fingerprinting from a scientific curiosity into a practical, everyday tool for justice. Their dedication and innovation inspired a new generation of scientists and law enforcement professionals. Their remarkable contributions have left an indelible mark on history, shaping the evolution of modern forensic science and earning global recognition for their achievements. The history of DNA fingerprinting stands as a testament to the power of scientific discovery, showing how one breakthrough can transform the pursuit of truth and justice for generations to come. DNA databases enhance the effectiveness of DNA fingerprinting globally. The USA's CODIS and the EU's PRUM treaty exemplify different approaches. CODIS, managed by the FBI, connects local, state, and national DNA databases. It helps identify suspects by comparing crime scene DNA with millions of profiles. The PRUM treaty allows EU states to exchange DNA data for cross-border crime fighting. Unlike CODIS, PRUM is decentralized, respecting national sovereignty and privacy. These systems reflect different philosophies on data management and privacy. For UPSC aspirants, understanding these models is crucial for grasping global best practices. In India, DNA fingerprinting has grown since the late 1980s, led by CDFD in Hyderabad. CDFD provides DNA profiling services for crime investigation and other purposes. Despite its use, India's legal framework for DNA evidence has been debated. The DNA Technology Regulation Bill aims to create a regulatory framework, but faces criticism. Concerns include privacy, data misuse, and potential surveillance. The Supreme Court has intervened, emphasizing consent and privacy in DNA collection. Judicial guidelines provide a temporary framework, while legislative debates continue. This highlights the tension between security and citizens' rights. Understanding these issues is vital for UPSC aspirants to navigate the complexities of DNA technology in governance. DNA fingerprinting's applications extend beyond criminal justice. Its accuracy makes it the gold standard for human identification. It links suspects to crime scenes and identifies disaster victims. DNA testing resolves civil disputes, like parentage and immigration cases. In medicine, it diagnoses genetic disorders and aids organ transplantation. In conservation, it combats poaching and monitors endangered species. DNA fingerprinting's versatility impacts governance, society, and the environment. For UPSC aspirants, understanding its wide applications is crucial. It demonstrates how scientific innovation influences various aspects of life. DNA fingerprinting, despite its benefits, faces challenges and risks. Sample contamination can lead to mixed profiles, complicating investigations. Degraded samples may yield ambiguous results, limiting their usefulness. High costs can restrict DNA testing to high-profile cases, creating inequality. DNA evidence interpretation is complex. A match doesn't prove guilt. Ethical concerns include privacy, consent, and data security. Large DNA databases risk creating a surveillance state. Security breaches could expose sensitive genetic data, leading to discrimination. Bias in DNA collection could reinforce social inequalities. These challenges require robust legal safeguards for responsible use. Addressing these issues ensures DNA technology is used justly and ethically. DNA fingerprinting is evolving with new technologies like rapid DNA analysis. Portable machines can generate profiles quickly, speeding up investigations. AI and forensic genealogy offer new ways to analyze complex DNA mixtures. These advancements raise privacy concerns, requiring clear ethical guidelines. For UPSC aspirants, DNA fingerprinting intersects with key syllabus areas. It exemplifies biotechnology's impact on governance and society. Understanding its scientific, legal, and ethical dimensions is crucial. It encourages critical thinking about regulatory frameworks for new technologies. This knowledge prepares aspirants for the challenges of modern administration.